Hello everyone, and welcome to another recorded lecture of BIO101 online. Today we will discuss genetics and inheritance. Before we move any further, I'm going to highly suggest making flashcards. There are a lot of different vocabulary terms, and it's easy to get confused between a lot of those terms. So having flashcards or downloading the Quizlet app on your phone will make it a lot easier for you to study for Chapter 11. So just to review, last chapter we discussed meiosis, which spoke about how germ cells undergo meiosis, which divides into meiosis 1 and meiosis 2, and that gives us the genetic variation that we're going to talk about today. So this is where we're going to bridge off of. We spoke about meiosis in Chapter 10. Now we said that meiosis is important so we get genetic variation. So today we're going to talk about how this genetic variation can be passed on from generation to generation. We said that somatic cells are diploid. They have two sets of homologous chromosomes, where haploid cells, like gametes, only have one set of chromosomes. And gametes that include sperm cells and egg cells combine to form a diploid zygote. So that should be the information that's clear to you before you move forward. So let's now talk about this genetic variation, and let's come up with a few terms of what genetics is all about. So what we're going to talk about today is, why do some children look like only one parent, while others look more like their great-great-grandparents? What causes similarities and differences of appearance, and what causes the skipping of generations? Right, so those are some of the questions we're going to try to address in this lecture. And let's introduce some terms. So a gene. A gene is a segment of DNA that carries biological information. That information is usually instructions on how to make a certain protein. So we have many genes. Most of those genes specify on how to make a certain protein. And we know that proteins are required for all cellular functions, right? Proteins do the work of the cell. So proteins are like the hardware, and the genes, the DNA, is like the software. Genes are inherited from generation to generation, right? So with sexual reproduction or asexual reproduction, you're passing on your genes from generation to generation. And all living things need to be able to do that. So genetics is the study of inheritance patterns and variations in organisms. So a geneticist would study the different inheritance patterns and would study the different variations in organisms and would see how they appear and reappear for generation to generation. So geneticists specifically would track the inheritance of traits. And a trait is a physical, heritable characteristic. Heritable means able to be passed on. So an example of a trait would be curly hair. Right? That's a trait. It's a physical, heritable characteristic. So a geneticist might be interested in seeing, how does the trait of curly hair get passed on from mother to daughter and mother to son, etc. Traits are determined by the specific alleles that an individual has. And we said that alleles are different versions of a particular gene. So we all have two copies of the hair texture gene. This is simplifying it, right? We both have a hair texture gene. We got one from mom and one from dad. But it can be of two different alleles, right? Each gene can either be a straight hair allele or a curly hair allele. So depending on the two alleles, right, the trait might be curly hair or straight hair. So here are some examples of traits. Not all of these are controlled by only one gene, I will warn you. So it's a little more complicated than I'm making it seem. But I'm trying to introduce the idea that traits are determined by alleles, which are just flavors of genes. Right? In diploid cells, like somatic cells, we have 2 alleles for each gene that are located at a particular locus of homologous chromosomes. A locus is like a location. So a locus is a precise location on a chromosome, and we know that each gene has a specific locus. So in homologous chromosomes, you're going to find different alleles for a particular gene at the same locus, at the same location. So we know that genes exist in different versions called alleles. Let's say we're looking at a gene for seed shape, the shape of a seed. One allele might encode for round seeds. A different allele of the seed-shaped gene might encode for wrinkled seeds. Right? But since these are both alleles of the same gene, they're going to be at the same locus of homologous chromosomes. So this chromosome came from mom, this one came from dad. Right? This is both, let's say this is chromosome 1 from mom, chromosome 1 from dad. You're both going to have the same gene in the same location, but the allele can be different, right? One could encode for round seeds, one could encode wrinkled. Just like one could, if it's the hair gene, one could encode for straight hair, one could encode for curly hair. So I'd like you to review this video, which is in the Connect ebook. And this talks about a recap of what our chromosomes look like in a zygote, talks about sister chromatids versus homologous chromosomes. It also discusses how these alleles can be the same or different on homologous chromosomes. If you have any confusion about alleles, sister chromatids, homologous chromosomes, if there's any confusion, if it's not 100% clear, watch this video until it is clear. It's going to be very hard to understand genetics if you don't understand meiosis. So please review this video on homologous chromosomes. It's actually also in the homework assignment. So just to review, we said that diploid cells have 2 alleles for each gene, and those are at the same genetic loci. Right? Loci is plural for locus. So here we have duplicated homologous chromosomes. This is a homologous pair of chromosomes. One came from dad, one came from mom. And you have, again, homologous chromosomes have the same genes at the same loci. So here's the gene A locus. So both mom and dad are going to give the big A allele for gene A. At gene B locus, we see that dad is giving the little b allele, where mom is giving the big B allele. So whereas big B and little b are the same gene, they're different alleles, they're different versions of that gene, right? So in the case of B, we see that there are different alleles of gene B at that locus, at the gene B locus. So the genotype, that's an important term. Genotype refers to the set of alleles that an individual possesses. So the genotype refers to the set of alleles that an individual possesses. If the 2 alleles of a gene are the same, we say that genotype is homozygous. Right? Remember, we have 2 diploid cells have 2 alleles for each gene. If those 2 alleles are the same, the person is homozygous for that gene. So that's the case of big A. This individual is homozygous for big A because both mom and dad are giving this person a big A. If the 2 alleles of a gene are different, we then say that genotype is heterozygous. So this genotype would be big B, little b, right? Mom and dad are giving different alleles. So in this example, this individual would be homozygous for gene A, but heterozygous for gene B. So just to review, right, we have 2 alleles for each gene. If you have a homozygous genotype, that means both mom and dad gave you the same allele. If it's a heterozygous genotype, that means mom and dad gave you different alleles. And alleles are usually abbreviated with letters. So like B, And this shows that big B means this is homozygous. If mom gave you a big B and dad gave you big B, let's say that encodes for brown eyes, right? This means you're homozygous for brown eyes. If mom and dad were both blue-eyed, you could be homozygous for blue eyes as well, little b, little b. But if mom had brown eyes and dad had blue eyes, then you would be heterozygous, big B, little b. So, here is another picture taken from the book. This is a different chromosome that has 4 genes, G, R, S, and T. Now, these are homologous chromosomes that have the same genes at the same specific loci, but the alleles might be different. So after replication, before meiosis 1, we then have each homologous chromosome has a sister chromatid. Right? And what happens in meiosis 1? Right, so think about that. What happens in meiosis 1 with these two homologous chromosomes? Right? So there's crossing over. Crossing over occurs. So think about what that could result in. Right? Crossing over can occur right here, for example. So before I get to that, I want you to tell me, is this individual homozygous or heterozygous for each gene? So for gene G, is it homozygous or heterozygous? For gene R, homozygous, heterozygous. So you should tell me that right now. And you should be telling me that it's heterozygous for everything. Right? For each gene, G, R, S, and T, heterozygous for anything. But my question for you is it possible for a gamete made by this individual to have the alleles little g, little r, little s, and little t? The answer is yes, right? Because if crossing over occurs between genes S and T, it's possible to get a chromatid to have little g, little r, little s, and little t. So this chromatid would look all blue, but would have this red bottom tip on it. So it's possible for a gamete made by this individual to have all lowercase letters if crossing over were to occur. So I hope that makes sense, right? That's crossing over, we mix and match alleles. So read this question and pause here. Right, so the answer is the 2 chromosomes are homologous because they have the same genes in the same location. So we said that genotype refers to the alleles of an individual. Phenotype refers to the physical appearance of the individual. So phenotype is a new word that's very important. Right, so phenotype is encoded by the genotype. So the phenotype is the physical appearance, which is largely determined by which proteins are made by the genotype. Right? So the genotype encodes for the phenotype. The genotype is in our chromosomes, right? Those are the alleles. Those alleles will specify which proteins get made, which will determine the phenotype. Right? This is explained by the central dogma of biology, which you might be familiar with. We'll talk a lot more about that in class, but the central dogma of biology tells us that DNA encodes for proteins. So the genotype, the DNA encodes for proteins of the phenotype. However, genes are not the whole story, right? The phenotype is actually heavily influenced by the environment and other epigenetic factors. Epigenetic means outside of genetics. And we'll talk about epigenetics in chapter 13. There's a whole lot of things going on that influence phenotype other than just genotype. And that's something that's become more and more obvious as we continue to study genetics. So, we said that the inheritance of alleles, genotype, determines physical traits, phenotype. So suppose we have a homozygous brown-eyed individual. The genotype would be big B, and the phenotype would be brown eyes. the homozygous blue-eyed individual would be little b, and would have blue eyes. What about if mom and dad each gave a different allele, and you had a heterozygous big B and little b? What would the phenotype be? Right? The connection is not always clear, because we would see that this individual has brown eyes. Even though there's a blue-eyed allele, this individual's phenotype is still brown eyes. So this is interesting, right? The connection between genotype and phenotype is not that clear-cut. And this is what was revealed by early geneticists in the 19th century. So up until the 19th century, there were two very popular theories of inheritance. They were very misleading. One was called the homunculus. And that idea was that offspring are a result of just one parent's contribution. Namely, they said that the father's sperm contained a mini human in it. And then when it would go into the female would just allow that mini human to grow into an embryo. So many microscopists actually said that they saw a fully formed miniature fetus in the sperm's head. And they were convinced that the sperm has this mini little man in it, mini little female in it, and that it just develops into a bigger thing once it's in the female. So we know that's nonsense. Another theory that was a little more logical, I think, was that parental traits become mixed, creating offspring with like an intermediate. So for example, if a tall dad and a short mom got together, they would have intermediate children. If a black bird and a white bird had children, they would all be gray. But this doesn't really make sense, blending inheritance, because that means traits would always become less extreme over time. Right? Everything would just be more blended until everything is just gray and kind of an equilibrium. So that's not the case, right? We still have black birds and white birds, and we have tall people and short people. They're still, not everything is just blended over time. So it wasn't until Gregor Mendel's experiments disproved this idea in the 60s that we began to understand how inheritance actually works. So Gregor Mendel is the father of genetics. He was actually a monk who lived in a monastery, and he was way more of a mathematician than a biologist. So in the 1860s, prior to the 1860s, he studied the inheritance patterns in pea plants. So peas were really easy to grow. They have a short generation time, and Mendel had a lot of time to count peas. So he looked specifically at inheritance patterns of discrete traits. What this means is he chose traits that were very easy to observe and that were objective. So he looked at pea plants that were either green or yellow, tall or short, right? Round or wrinkled. These are discrete traits. It's either or. Very objective. It's not really up for interpretation. So by simplifying it in this way, he was able to do a lot of controlled experiments that were very simple, but powerful, right? That had a very powerful conclusion. So by studying these discrete traits, he was able to conclude a lot about how genes are transmitted from generation to generation. So his paper, Experiments in Plant Hybridization, was published in 1866 and became the cornerstone of modern genetics. This was before we even knew what a gene was. What Mendel really did was he inferred laws of genetics. He came up with two big laws of genetics that allowed predictions about which traits would appear, disappear, and then reappear from generation to generation. What Mendel really did in the end was propose that inheritance involves the reshuffling of particular units from generation to generation. It's not just a blending from generation to generation. There are these units that dictate our phenotype that get shuffled between generation to generation. We now call these units that get reshuffled genes. Right? He didn't know what genes were. So he called it the particulate theory of inheritance, because there are these particular units that get mixed and matched during the process of development. So Mendel didn't even need to use a microscope for a lot of his work, but this was a microscope used by Mendel. This was his paper that he published. And like I mentioned, he used pea plants. And a pea plant is like any other flower. The flower has both female parts, it has ovules, which are like the eggs, and it has a male part, that has anthers that have pollen, which is the equivalent of sperm. So that's important to know. Ovules are like eggs. The pollen from anthers are like sperm. So flowers have male and female parts. And this is a picture of peas, what they really look like. You might have seen a pea plant before. Right? So these are the pea flowers. So it's important that Mendel only used true breeding pea plants in his experiment. True breeding is the same thing as saying purebred or pure breeding. This means these parental plants always produced identical progeny to themselves. They were purely bred. There is no, nothing hybrid about them. So he used plants that would, for example, if he took a green plant, a green pea plant, and mated it with itself, it would only give green plants, right? Because it's true breeding. So that's really important that he started off with true breeding plants. So, right, they're homozygous for a given trait. And it's important to be familiar with how plants can be fertilized. So plants can fertilize themselves, meaning pollen from the anther of this flower can fertilize an ovule of the same flower. That's called self-fertilization. So make sure that's clear. Or you can do cross-fertilization. That's when you want to mate two different flowers. So when we're crossing, when we say we're mating flowers or crossing, we're doing cross-fertilization, which means we're taking pollen from one flower and putting it on the stigma, the female part of a different flower that had its male parts removed. Right, so cross-fertilization, you're taking the pollen of one flower, putting it on the female part of a different flower that had its anthers removed. That flower will then develop a fruit, which is the peas. And then you can plant these peas in the ground to get the next generation of plant. This is a little confusing, and I'll admit that when I first learned about Mendel and his pea plants, I didn't fully conceptualize how to mate these peas and how to cross-fertilize and how you would get peas that you can plant to get the next generation. To understand it, I recommend watching this video on YouTube, where you can see somebody actually crossing pea plants so you can understand how this works. So plants can fertilize themselves or can be cross-fertilized. And the offspring of some pea plants gave Mendel some clues about the genes of the parents. Right? He said that not all pea plants were true breeding. So he only wanted to use true breeding plants because they produce offspring identical to themselves. But not all plants are like that. For example, a true breeding green pea plant, that when you self-fertilize it, it'll only produce green offspring. Same with a yellow pea plant. If you self-fertilize a yellow pea plant and only produces yellow peas, we say that's true breeding. If you plant a yellow pea and you get a mixture of seed colors, that means this plant was a hybrid. This is not true breeding, and you would not be able to use this plant in a mating experiment. or Mendel would not have used this in his original mating experiments, I should say. So Mendel only stuck with true breeding plants. Okay, so now let's talk about Mendel's experiments. So Mendel crossed true breeding pea plants that differed in only one trait. This means the pea plants were identical in every possible way except for just one characteristic, which was first seed color. So he took two parental plants. He took pure breeding yellow peas and pure breeding green peas, and he mated them together. What this literally means was he took the pollen from a flower of a yellow pea plant and he put it on the stigma of a green pea plant that didn't have any anthers. Then he saw the flowers, he saw the new peas produce, and then he looked at the color of the peas that grew from those flowers. That's the next generation. And then he can plant those peas again to see what it grows into. So let's go back to the beginning. We're going to start over here. He took two parents. We call the parents the pea generation, the parental generation. He took yellow peas and mated them with green peas. Right? And the pea is the parental generation. We always say that the parental generation is the pea generation. Not to be confused with peas and pea. That's a coincidence. So Mendel's pea generation is yellow peas, the male part, green peas, female part. After they were crossed, he examined the phenotypes of the offspring peas. We call the first generation the F1, or the first filial generation. Right? Filial means like sons and daughters. So the first filial generation, F1, are the children of the parents. So it always goes P and then F1. So hypothesize the phenotype of the F1 peas. What do you think the F1 peas would look like? So he saw that the F1 peas were all yellow. And this is really interesting. He's like, okay, where did the green peas go? He thought maybe it would be like a light green color or a combination of green and yellow. But he saw only yellow peas. So that was interesting to him. When the F1 peas, these yellow peas, are planted, they could be grown into mature pea plants that have flowers. You can then self-fertilize those flowers. So you're taking the male part of the flower and putting it on the female part of that same flower and wait for the peas to grow. These next generation of peas would be considered the F2 generation, the second filial generation. This is important, right? The F1 peas are crossed to each other. Right, so the F1 peas are planted, and those flowers that grow from that pea plant are self-fertilized to generate the F2 generation. So it always goes, P is the parent, they have the first filial generation, the F1, and then the F1s are mated to themselves to get the F2 generation. When two F1s are mated to get the F2 generation, that's called a monohybrid cross. Why is it called a monohybrid cross? Because you're crossing two hybrid organisms, right? Two F1, they're hybrids, that differ only in a single trait, which is the pea color. So this is a monohybrid cross. When we take two F1s, they're both hybrids, and we're crossing them together to get the F2 generation. So now hypothesize the phenotype of the F2 peas. He saw something very interesting in the F2. He counted a total of about 8,000 peas, and he noticed that there was a very specific ratio of 3:1, yellow to green. Another way to say 3:1 is 75% were yellow, 25% were green, right? Or 3/4 were yellow, 1/4 were green. That's what a 3-to-1 ratio means. So after this kind of experiment, Mendel concluded a few things that are very important. He said, when you cross two pure breeding plants that differ in one trait, the F1 progeny will only have one of the parental traits. He then saw both parental traits reappear in the F2 progeny in a 3-to-1 ratio. He concluded that some traits are either dominant or recessive. These are two important terms. The trait that appears in F1 is dominant, and the trait that disappears in the F1 is recessive. So in the case of peas and pea color, which is dominant, yellow or green? Right? Yellow is dominant over green, right? Because the F1 generation had all yellow. So yellow is dominant over green. The yellow is able to mask the expression of the green color. But just because a color is lost in one generation, it doesn't mean it's not going to reappear in the next generation, right? So some traits skip a generation. Mendel performed reciprocal crosses, meaning he not only took the pollen from yellow peas to fertilize the eggs from green peas, he also took the pollen from green peas to fertilize eggs from yellow peas, and he saw the same exact thing. So the results from reciprocal crosses produced similar results. This disproved blending hypothesis, right? You're not blending. There must be specific things in these F1s that get reshuffled in the next generation to give us an assortment of colors, right? When it wasn't there in the generation before. So there must be units of inheritance. And Mendel didn't stop there, right? He actually looked at 7 different pairs of discrete traits that were either or. So he looked at seed color, seed shape. Was it yellow? Was it round or wrinkled? Was the flower color purple or white? Was the stem short or long? Was the unripe pod color green or yellow, etc.? So he looked at 7 different traits, and he saw very similar results. So now let's look at another example, and you should have a blank sheet of paper with you because we're going to go through this step by step. So in addition to the seed color of yellow or green, Mendel also looked at the pea plant height. So plant height is determined by 2 alleles. Big T is tall, little t is short. What are the genotypes and phenotypes of each parent? Right? So of the pea generation, this individual's genotype is big T, big T. We could say that this is homozygous dominant. Right? I'll introduce dominant and recessive again in the next slide. But these are both homozygous genotypes because they both have 2 of the same allele. Right? This is little t, homozygous, big T, big T. The phenotype of this plant is tall. The phenotype of this plant is short. Right? So those are the genotypes and the phenotypes of the parents. So now let's try to visualize what's actually going on here. Each parental pea plant has to undergo meiosis to produce gametes. So what kind of gamete could be formed? Well, this plant has got 1 copy of big T from mom, 1 from dad. It only has 1 choice. It can only give a big T. Right? So in meiosis, when the alleles mix and match, if there's only big T's there to begin with, there's only going to be a big T given in the sperm or egg, or in the pollen or the ovule. Same idea over here. This is a homozygous pure breeding plant. It can only give a little t. So if I cross these two parents, what will the F1 look like? What will the F1 genotypes and phenotypes be? Right? There's only one option. They all have to inherit one big T from this parent and a little t from this parent. Right? They're two haploid gametes. They come together to form the F1 generation, which will be big T, little t. What's the phenotype? Right? Which trait is dominant? It must be tall. Right? Because tall is what appeared in the F1. The short phenotype disappeared in the F1, so that must be recessive. So now we have to figure out what would happen if I self-fertilized this F1 plant and then grew the peas. What are the possibilities? Right? So we're doing a monohybrid cross now. Remember, these are both hybrids, big T, little t, and they're only differing in one trait, in height. So I'm going to self-fertilize these F1 peas, and now I want to figure out what are the possible offspring. What are the possible plants going to look like in the F2 generation? Okay, so that's the question we're trying to ask. So after meiosis, the F1, the gametes of this plant can be either big T or a little t. So let's think about this. This plant has sperm and pollen and eggs. Since sperm and egg are haploid, it's either going to have a big T or a little t. Right? So the eggs of this plant will either have a big T or a little t. The sperm of these plants will have either a big T or a little t. So what are the possible ways that these big T's and little t's can come together to form the zygote? Right? To determine the possible F2 genotypes, we make a Punnett square. And you might have done these before, right? But a Punnett square shows you the probable offspring that can result from a mating of individuals. In this case, this is a self-fertilization. So the eggs will be big T, little t, and the sperm will be big T, little t. Right? But we don't know. This big T could come together with this big T, right? And that would mean that we can get a big T plant. One that's homozygous dominant. Right? Dominant is always capital letter. It's also possible for a big T sperm to unite with a little t egg. Right? So that's another possibility. It's also possible for a big T egg to unite with a little t sperm. Or it's possible for a little t egg to unite with a little t sperm. Right? So these are all possibilities. So what we're doing in these kinds of monohybrid crosses or in a Punnett square is we're lining up the different genotypes of the eggs, right? The different alleles that the egg can have, the different alleles that the sperm can have, and then we're seeing how they could come together, right? How can different eggs and different sperm unite? What are the probable F2 genotypes going to be? So what he saw was a consistent ratio of offspring from a monohybrid cross. He saw a genotype ratio of 1 to 2 to 1. What that means is, whenever he did a monohybrid cross, which like big T, little t, crossed with another big T, little t, he saw that there was, out of every 4 pea plants, 1 out of 4 was homozygous dominant, big T, 2 out of 4 were heterozygous, and 1 out of 4 was homozygous recessive. That was a consistent thing that he saw. He saw a consistent phenotype ratio of 3 to 1, whereas 3 out of 4 showed the dominant phenotype, 1 out of 4 showed the recessive phenotype. So he saw that 75% of the plants were tall, 25% of the plants were short. So I want to just make sure that you know how to draw a Punnett square. Right, so you just line up the different sperm genotypes on one side, egg genotypes on the other, and then you're looking at the possible ways that sperm and egg can recombine to form different zygotes. And in the case of a monohybrid cross, we see a genotype ratio of 1 to 2 to 1, phenotype ratio of 3 to 1. So you should remember those. And it's easy to just do it out if you forget. So this brings us to Mendel's first law, which is the law of segregation. Right? Punnett squares basically just summarized meiosis and fertilization. And this is all explained by Mendel's law. So Mendel's law says that each individual has a pair of alleles for each trait. Right? We're diploid. We have 2 copies of every gene, 2 different alleles. Those alleles segregate. Right? They separate during gamete formation. We know in meiosis 1 that, mom and dad's homologous chromosomes line up, they do crossing over, and then they separate to 2 separate cells. That's what Mendel figured out before we even knew what was going on in meiosis. He knew that these alleles can segregate when sperm and egg are formed. Right, so for example, if I have a yellow pea plant, that's big Y, once it grows into a plant, it can only make big Y gametes. So all of its sperm will have big Y, all of its eggs will have big Y. Little y green pea, pure breeding plant, you could grow that into a plant, and that plant will only make little y gametes. Therefore, right, each gamete only has 1 allele from each pair of alleles. Right? You're going to have only 1 allele of each gamete. And 2 gametes, 1 from each parent, unite at random during fertilization. So if I have a big Y sperm and a little y egg, right, those will get fertilized. And that would give me a zygote, which has 1 of each allele, big Y, little y. And if I were to plant that seed, right, I would get the F1 hybrid that would still look yellow because big Y is dominant to little y. Right, so the law of segregation says that alleles separate during gamete formation. So each gamete only has 1 of each allele. But then when 2 gametes come together, we get a zygote that has 2 copies of each gene, right? 2 alleles for each trait. Let's do another example. Suppose we take pea plant and we're looking at the color of plants. So purebred purple flowers were crossed with purebred white flowers. What do you think the F1 generation will look like? They actually saw only purple. So which is dominant? Is purple dominant or white dominant? Right? Purple must be dominant, right? Because the white disappeared. So the F1 offspring were self-pollinated to create the F2. And we see that 3-to-1 ratio once again, where 75% of the F2 flowers were purple and 25% were white. So the white flowers reappeared after skipping generations. So now I want to show you how we can do Punnett squares to illustrate all of this. And with dominant, right, we always write the dominant allele in capital letters. Lowercase is always the recessive. Right, that's just how we always draw it out. So I'm going to try to let's draw this Punnett square. Let's say big P is purple. So big P is purple. I'm going to try my best to, that's pretty terrible. and little p is going to be white. Okay. You kind of get the idea. So what we're going to do here is show the parental cross. Let's show a parent purple plant mating with a white plant. So what are the genotypes of each of these parents? Right, this would have to be big P, because it's pure breeding in purple. This one would have to be little p, little p. I don't like using p's because they look the same when you draw them out. But for this example, we'll just try to bear with it. So in your notes, draw a Punnett square that shows the crossing of these two flowers. And I'm going to draw one in the meantime. So let's draw the Punnett square. And on top, we'll put the two alleles that the purple flower has. So big P and big P. The white flower has a little p and another little p. So in these boxes, right, you can draw, if you like, you can draw more boxes like that. That's easier to see. And now in the boxes, we're going to put the different possibilities for the F1 genotype, right? These are the parents, big P, little p, little p. All of the F1 will get a big P from the purple and a little p from the white. Therefore, every single F1 will have the same genotype, which is big P, little p, which are all heterozygous. And what phenotype would big P, little p be? It would be purple. And that's exactly what we saw here. So the genotypes of all these purple flowers here are big P, little p. This was little p, little p. This is big P, big P. So now I want you to show me, so you should pause here, show me the cross that generated these F2. I want you to show me a Punnett square that illustrates the mating of two F1 flowers, self-fertilization of an F1 flower, to generate the F2 offspring. So you should pause here and draw a Punnett square. Right? So we're doing the mating of two heterozygotes. So this time we're doing big P, little p, times big P, little p. So what we're going to get in this mating is one big P, one big P, little p, one or another big P, little p, and finally little p, little p. These are the four possible combinations of genotypes we can get, combinations of alleles. And what would a big P phenotype be? Purple. What about big P, little p? Still purple. What about this big P, little p? Purple. What about this little p, white. So 3 out of the 4 will be purple. 1 out of the 4 will be white. This is exactly what the 3-to-1 ratio is saying. 75%, or 3 out of every 4 will be purple in a monohybrid cross. So big P, little p times big P, little p is a monohybrid cross. So make sure you're able to draw Punnett squares that depict a monohybrid cross. So Mendel looked at stem length, seed color, and flower color, but also 4 other traits, and the F2 results were consistent. Out of 20,000 different specimens, he saw that there was a 3-to-1 ratio in a monohybrid cross of the F2 offspring. Right? So when you mate 2 hybrid organisms together, you get a 3-to-1 ratio in the F2 generation, where 3 out of 4 show the dominant phenotype, 1 out of the 4 show the recessive phenotype. So the dominant allele masks the expression of the recessive allele. Right? So, and we said the dominant allele is always a capital letter, the recessive allele is always the lowercase letter. And the P gene, the P gene controls flower color in pea plants. Right? We said big P encodes for purple flowers, little p encodes for white flowers. Since purple is dominant to white, either big P, or little p, or big P, little p genotypes would give us the purple color phenotype. Right? So why does this make sense, right? Why does big P, or big P, little p give you purple flowers, whereas little p doesn't? Well, let's think about this, what this really means. The gene for flower color, big P, encodes for a functional pigment-making enzyme. So when you have a functional copy of gene P, you can make purple pigment. Right? The dominant P allele is active and produces purple pigment. Since purple pigment is the most common in the wild, most flowers are purple, this is called the wild type. Purple is called the wild type. The recessive little p allele is inactive. It's mutated for some reason, so no pigment is produced. Recessive alleles are usually loss of function, right? It's a result of some kind of mutation. So when you have a little p allele, no purple pigment can be produced. And that's why the flowers would look white. Right? So flowers with no pigment are white. They have little p. But even if you have just one copy of big P, right? So a big P, little p has only one copy of the pigment-making enzyme. That's enough for it to look completely purple. Right? So since purple is completely dominant to white, having just one copy of big P is enough for the entire plant to have purple flowers. Right, so I hope that makes sense so far. Right? So the little p means there's no functional copy of the enzyme that makes the purple pigment. So it was just white because it had no pigment at all. So let's just test your knowledge so far. Which of the following is a homozygous recessive genotype? The answer is C. In flying monkeys, gray fur color, big G, is dominant to white fur, little g. Which of the following genotypes would lead to a gray phenotype? The answer is D. Either A or C would lead to a gray phenotype. Which would lead to a white phenotype? So pause here, and the answer is B. Little g. If a purebred gray monkey were mated to a heterozygous gray monkey, what percentage of their offspring would be gray? So this is a Punnett square problem. If a purebred gray monkey, so let's take that step by step. What is a purebred gray monkey? Well, we know that gray fur color is big G, and we know pure breeding means it must be two of the same. It must be homozygous. So on top, we're going to put big G. And it's mated to a heterozygous gray monkey. So what is a heterozygous gray monkey? That's big G, little g. So and now it's asking if these two were mated, what percentage of their offspring would likely be gray? So if I were to fill in these boxes, I would get big G, another big G, big G, little g, and then big G, little g. What percentage of their offspring would be gray? 100% would still be gray. If a white monkey were mated to a heterozygous gray monkey, what offspring would be gray? So white monkey would be little g, little g. Heterozygous gray monkey would be big G, little g. So let's fill in the Punnett square. And what we see here is that 50% will be big G, little g, which would mean they would be gray. 50% would be little g, which means they'll be white. So the answer is B, 50% will be gray. So this brings us to the next topic called a test cross. So far, right, we said that a homozygous recessive genotype, like little p, gives you the recessive phenotype, right, your white flowers, right? But for an individual with a dominant phenotype, the genotype is not as clear, right? If we have a purple flower, how do we know if it's homozygous for purple or heterozygous for purple? Right? It's not as clear, right? A purple flower can be big P, or big P, little p. So a test cross can be used to determine the genotype of an individual that has the dominant phenotype, right? Somebody with a dominant phenotype can be homozygous, dominant, or heterozygous. So we have to do a test cross to determine the genotype of that unknown individual. The way a test cross works is you cross the unknown dominant individual with a homozygous recessive individual and then look at their offspring. So let's do an example with the flower. Let's say we have a purple flower. How can we determine the genotype of this purple flower? It could be big P, or big P, little p. What we have to do is mate this unknown flower with a homozygous recessive white flower plant. If we mate this with a white flowered plant, right, that's what we have to do. We'll mate it with a white flowered plant. If it's big P, what would you expect the progeny to look like? If it's big P and you mate it with little p, what would you expect the progeny to look like? Right? So if you do this mating, you mate the purple flower with the white flower. Suppose you see they're all purple. That means this purple flower was homozygous dominant, right? Because all of the offspring were still purple. If you mated the purple flower to this white flower and saw 50-50, that means that the purple flower must have been heterozygous, because it was able to give that little p allele. So 50% were still white. So make sure that makes sense. Like, make sure you understand how a test cross works. You cross a purple flower with a white flower and observe 100% purple offspring in the F1. What was the genotype of the purple parent? So pause here, and the answer is A, big P. So if you observe 50% purple offspring in the F1, what was the genotype of the purple parent? So pause here, and the answer is B, big P, little p. So now we'll do two practice problems. So you can pause here and do this problem now. Say in pea pod color this time, I'm saying green is dominant in different species of pea, let's say. Big G. And yellow is recessive, little g. And you take a yellow father and a true breeding green mother, and you mate them together. So what are the genotypes of the parents, of these two parents? What colors are the children? And what percentage of the F2 will be yellow? So again, this is different species of pea. We're not looking at big Y and little y. This time, green is dominant. And doing genetics problems has everything to do with just reading comprehension. It's the most important thing for genetics, to just read the problem step by step and underline the important information. right? Every problem might be different. So this is saying big G is dominant over little g, which is yellow. So pause here and try to figure out the answers to A, B, and C. So a yellow father must be little g, and a true breeding green mother must be big G, because she's true breeding, must be 2 of each. What colors are the children? Well, all of them would be big G, if you did the Punnett square. All be hybrids. And the color would be green, since green is dominant, according to the problem. So B is green. What percentage of the F2 will be yellow? Well, for that, we have to do a Punnett square. Big G times big G. And let's do the Punnett square out. Oops, oh, that's an ugly G. But here we have big G, big G, big G, little g, big G, little g, little g, little g. What percentage of the F2 will be yellow? Only the little g will be yellow, which is 25%. So 25% will be little g, which is yellow in this case. So now I want you to try one more example. So you can pause here and solve this one. So if you have a heterozygous father for pod shape, so smooth pods are dominant over bumpy pods. You have a heterozygous father, that must have been a smooth pod father, and a bumpy pod mother. All right, the genotypes must be big S, crossed by little s. What shapes will the children be? So let's do a Punnett square. Again, this is not the best one to do it. So maybe I'll change this in the slides, actually. But you have the idea. So big S times little s would give us 2 big S, and 2 little s. So the question asks, what shapes are the children? They will be 50% bumpy and 50% smooth. So make sure you can do these kinds of problems. There's the interactive Connect assignment that has multiple kinds of these questions, and they actually keep on changing. So you can repeat them as many times as you want and do different forms of each question. So before you move on, make sure you understand how to do these basic kinds of problems. So now let's talk about some dominant and recessive traits in humans. So you might have to look in the mirror, but are your earlobes attached or detached? Right, so it's possible to have detached earlobes, right, where the lobe is distinct and not attached to the side of the face. And if big E encodes for detached earlobes, and big E is dominant over little e, what would the heterozygous phenotype be? It would be detached as well, right? So big E is homozygous dominant, 2 capital letters, would be the detached phenotype. Heterozygous, big E would be detached as well, whereas little e is recessive, that would be attached earlobes. Is it possible for 2 parents with detached earlobes to have a child with attached earlobes? So think about that for a second. Is it possible? The answer is yes. The only way that would be possible is if both parents were heterozygous. So try to draw a Punnett square that shows 2 detached earlobed parents having an attached earlobe child. So a question on an exam could be 2 heterozygous parents for detached earlobes have a child. What is the likelihood that the child will have attached earlobes? For that, you would have to draw a Punnett square. So let's do it together. We'll start by calling, let's use big E, little e is one parent. And big E, little e is the other parent. So we're going to have one that's big E. This will be big E, little e, big E, little e, and little e, little e. 1 out of 4 children would theoretically be little e, which would have attached earlobes. So this means there's a 25% chance that 2 heterozygote detached earlobed parents would have a child with attached earlobes. It's 25% chance, or 1 in 4. Let me just clear the pen, and you can see the exact same Punnett square, that you would have 25%, or 1 out of 4, attached. So now let's transition to talking about diseases and disorders. So phenylketonuria is a human genetic disorder, meaning that it's a result of a gene deficiency. There's something wrong with the genes. So phenylalanine is a normal amino acid found in many foods, one of the 20 amino acids required for life. And we need to metabolize phenylalanine with a special enzyme called phenylalanine hydroxylase. So most of us have this enzyme that breaks down phenylalanine, and that's great. If you have excess phenylalanine, that's really bad. That can cause nervous system disorders and a lot of complications down the road. So you don't want to have excess phenylalanine. It's important that we have this enzyme, phenylalanine hydroxylase, always getting rid of excess phenylalanine. Great. How many copies of phenylalanine hydroxylase do you have in each of your cells? You have 2, right? You got 1 copy from mom and 1 copy from dad. Well, a phenylketonuric person is somebody who has no functional copy of that enzyme, phenylalanine hydroxylase. So they have nervous system disorders, and it can be deadly. So this disease is a recessively inherited trait because the person has to have 2 copies of the recessive mutated gene to have the disorder. Right? We call it a recessively inherited trait because you need to have 2 copies of the recessive allele to have the disorder. Even 1 copy of the normal gene is enough to metabolize the phenylalanine. So for example, if we wanted to put it as genotypes, I would say that big P is healthy. Big P, little p is also healthy, right? And big P encodes for phenylalanine hydroxylase in this example, right? This person would have 1 gene with a good copy of the enzyme and 1 gene with a mutated copy. Somebody with phenylketonuria would be little p, which means both copies of their phenylalanine hydroxylase gene would be defunct. They would be mutated. Therefore, they can't metabolize phenylalanine. You might have seen on Diet Coke, it says phenylketonurics contains phenylalanine, right? This is a warning for anybody with phenylketonuria to not drink Diet Coke, right? Because that is aspartame, which is partially phenylalanine. So babies are genotyped very early on. Actually, right when they're born, there's a heel prick. They prick the heel of the baby, and they can do some blood testing to see and to make sure that this baby has at least 1 copy of phenylalanine hydroxylase, and that the baby would not be a phenylketonuric, right? If the baby is a phenylketonuric, then it's possible to live a healthy life. You would just have to minimize consumption of phenylalanine and not have foods with a lot of phenylalanine. And a lot of people can live with this into adulthood. Just as a side note, the normal gene is referred to as wild type. So a mutant is different than wild type. A mutant gene is defunct. Or not always defunct, I should say. It has a different function than the wild type. So you can think about the normal gene as wild type. By definition, it's more than 1% of the population is wild type. And then if it's not wild type, it's a mutated version. So just note that, you should know that in your notes. Wild type in genetics means the normal or the most common type. So Punnett squares can be used to track the inheritance of genetic disorders like phenylketonuria and cystic fibrosis. Just like phenylketonuria, to inherit CF, cystic fibrosis, you need to have 2 mutant copies of this CFTR gene. So this is another recessive disorder. Let's call the healthy CFTR gene big F and the mutant gene little f. And we'll see what cystic fibrosis really is in the future, in a couple of slides. But again, having just 1 copy of the normal wild type gene, the big F, is enough to be healthy. So big F is healthy, little f means that's cystic fibrosis. If you're big F, little f, you're healthy, but you're a carrier. So what that means, you carry an allele for the disease. So this person is healthy, but some of his or her gametes will have this little f allele, which is the mutant CFTR gene that can cause cystic fibrosis. So a heterozygote is called the carrier in the case of diseases. So cystic fibrosis, we said, is caused by a recessive allele. If 2 healthy carriers have a child, what is the chance the child will be affected by cystic fibrosis? So you could pause here and try to figure it out yourself. I'm going to draw it right now, but please pause. So 2 carriers would be big F, little f, times big F, little f. Right? So the female is a healthy carrier. That means her gametes, her eggs, will either have a big F allele, which is the healthy copy of CFTR, or will have the mutant copy of the CFTR gene, little f. So half of mom's eggs have big F, half of mom's eggs have little f. The father is also a carrier. Half of his sperm have the big F, half of his sperm have the little f. So this Punnett square is saying, what are the possibilities of all sperm and all egg combinations? Well, it's possible that a big F egg and a big F sperm will meet. That will be a big F, healthy non-carrier. Right, big F, big. It's also possible that a mutant egg can unite with a healthy sperm, or a mutant sperm with a healthy egg, and that would give us a healthy carrier again. But if an unhealthy sperm, and mutant egg happen to meet, which will happen 25% of the time, then you will have a cystic fibrosis child. So here is a difficult question. A healthy female carrier for CF has a child with a male of unknown genotype. So we don't know what the male is, but we know that the female's genotype must be big F, little f. They have 8 children. 2 of the 8 children have cystic fibrosis. So you're asked, what was the phenotype of the man? So let's think backwards, right? Before you even look at the question, the answers, you should think about what makes sense to you. Not all of them have cystic fibrosis. In fact, not even half have cystic fibrosis. And we know if it's a person who has cystic fibrosis, you would get 50%. We just saw that in the last problem. If the man had cystic fibrosis, you would get 50%, right? These would be big little f, little f, little f, little f. And 50% of the children would have cystic fibrosis. But in this case, only 2 out of 8, only 1/4 has cystic fibrosis. Therefore, it would make sense if the male were a carrier as well. So if the male were a carrier and the female were a carrier, you would see a Punnett square, big F, big F, little f. And you would see that only 25% would be little f, and would have cystic fibrosis. So the phenotype of the man must be, he was a healthy carrier of cystic fibrosis. It must be B. So now I want to quickly go through what are called dihybrid crosses. And a dihybrid cross mates true breeding organisms that differ in two traits. This is different from a monohybrid cross. So for these, Mendel tracked each trait through two generations, the P, F1, F2 generation, right? So the P parent plants were true breeding plants that differed in two different traits. So for example, instead of just yellow peas being mated to green peas, he took yellow peas that were round and mated them to green peas that were wrinkled. So now they're differing in two separate traits, but they still breed true, right? So this round yellow pea plant can only produce more yellow round pea plants. When he mated round yellow peas with wrinkled green peas, he saw that the F1 plants showed both dominant characteristics. In this case, all of them were round and yellow. These were all hybrids, of course, right? You would imagine you could just look at these genotypes, and I'll show you this in the next slide. But if you were to take this yellow round pea plant, it can only give sperm that are big R and big Y, or eggs. This can only give little r and little y. So when those two come together, right, we're going to have a plant that's big R, big Y, little y. This is our F1. If we self-pollinate these F1 plants, we're going to get an F2 generation that has a very specific ratio of phenotypes. So whenever you do a dihybrid cross, there's a very specific ratio of phenotypes. And what a dihybrid cross is doing is essentially combining two different Punnett squares together. So a dihybrid cross shows a phenotypic ratio of 9 to 3 to 3 to 1 in the F2 generation. That is very, very important. 9 to 3 to 3 to 1 means out of every 16 pea plants, 9/16 will have both dominant traits, will be both round and yellow. Right? That's the 9. 3/16 will have one dominant trait but not the other. So 3/16 will be round and green, 3/16 will be wrinkled and yellow, and then 1/16 will be recessive for both. So 1/16 will be wrinkled and green. I want you to understand the ratios and how we got to them. So in this original cross, we took the two pure breeding plants. One was wrinkled and green, so little r, little y, and a male plant that was big R, big Y, big Y. And like I said, all these male sperm can only be big Y and big R. All these female eggs can only be little r and little y. So all the zygotes would have to be big R, big Y, little y, which would make them, again, yellow and round, because they have at least one copy of big R, at least one copy of big Y. And you would be told, of course, that round is dominant to wrinkled, yellow is dominant to green. In any problem, you would have to be told specifically which allele is dominant over which. So now let's do the F2 generation. We took two of these big R, big Y, little y individuals, and now we're mating them together. We're self-crossing them. But this part is not as easy, right? Because this pea plant can make 4 different kinds of gametes. This pea plant can make a gamete with big R and a big Y, a big R, a little y, a little r, a big Y, or a little r and a little y. You might be familiar with the term FOIL from multiplication, from algebra, F-O-I-L. and it stands for first, outer, inner, and last. And this is an easy way to think about how the gametes can form when you look at a dihybrid like this. What this means is this female of big R, big Y, little y can make 4 different kinds of gametes. They can be the first letters, so big R and big Y. It could be the outer letters, big R and little y. It could be the inner letters, little r and big Y. Or it can be the last letters, little r and little y. So when you look at this genotype, you can use FOIL to figure out the possible gametes. And what a dihybrid square is, dihybrid cross, is you're just looking at all the possible gametes, and then all the possible gametes, and then putting them together, just like we did in a regular Punnett square. But this time, each gamete, right, we're showing 2 different alleles. So again, since the male was the same genotype, you would have the same 4 gametes. So if this female egg were fertilized by this male sperm, you would get big R, big R, big Y, big Y. If this male sperm were fertilized by this gamete, we would get big R, big Y, little y. And so on and so forth. You'd be able to figure out the entire table. And you would see that 9/16, all of them that are shaded in purple, will be round and yellow. 3 out of the 16ths, those that are shaded in yellow, are round and green. You have these 3 over here will be green, will be wrinkled and yellow. And finally, 1/16 will be wrinkled and green. That's a dihybrid cross. Another way to look at it is using a branched diagram. So this is the same thing. This is showing the cross of the 2 F1s. In each, you can break down this into 2 separate crosses. We could look at first just the R gene, and then we could look at just the yellow gene, just the Y gene. So again, this is a little more information than you need to know for this class, but I'm going to just tell you the right way to do it. For the big R, for the R cross, you would have big R little r times big R little r, and you would see that 3/4, right, just by doing that Punnett square, big R, big R, little r, 3 out of 4 would still be round, and 1/4 will be wrinkled. The same proportion would be expected from seed color. 3/4 will be yellow, 1/4 will be green. You can then multiply. Again, you will not have to do this for the exam, but instead of making a big dihybrid square with 16 boxes, you can multiply the 2 probabilities. Right, so what is the chance of getting a round yellow pea? Well, we know that 3/4 will be round, and 3/4 will be yellow. 3/4 times 3/4 is 9/16. That's our ratio. That's the magic number from the ratio. How many will be round and green? Well, we know 75% will be round. 25% will be green. We knew that from here. So therefore, 3/16 will be round and green. Out of the 25% that were wrinkled, 75% will be yellow, which equals 3/16. And of those wrinkled, 1/4 of those will be green, which means 1/16. So by using 2 separate probabilities, we can put them together to come up with the dihybrid ratio of 9:3 to 3:1. So let's watch this video together. So actually, I'm going to have you watch this on your own. I just realized it's a 5-minute video. So instead of taking your time, please watch this video in the Connect ebook. It's a very good video that goes through an example of a dihybrid cross, and it really helps explain the ratios. So again, I want you to understand the ratios. Let's do one dihybrid cross practice problem. In cats, the allele for gray fur, big G, is dominant over the allele for beige fur, little g. the allele for solid coat, big S, is dominant over the allele for a striped coat, little s. If you have a pure breeding solid beige cat and you cross it with a pure breeding striped gray cat, what would you get? So pause now and try to figure out the genotypes of these two cats, right, and show the mating. Right, what are the parental genotypes? And state the genotypes and phenotypes of the F1 cats that would be produced as a result of this cross. So, if we have a solid and beige cat, and they're pure breeding, we know that it must be little g, because that's what encodes for beige fur, and big S is solid. And then we would get another cat, the striped gray cat would be big G, little s. When these cats mate, they would produce an F1 generation that are all solid and gray. Because this cat over here can only give one type of offspring, right? It can only give little g, big S, which is shown here. This cat over here can only give a big G, little s. Therefore, when these two sperm and egg come together, we're going to get all big G, big S, little s, which will be solid and gray cats. Now, calculate the phenotypes resulting from a cross between a pure breeding solid beige cat and one of these F1 offspring. So one of these F1 offspring is now mated with a solid beige cat. So pause here and try to do that in your notes. So I'm going to show you again how this is done. This solid beige cat can only give one type of gamete, right, which is little g and big S. However, this solid gray cat can give 4 different kinds of gametes, right? It can give a big G, big S, right over here. It can give a big G, little s. It could give a little g, big S, or a little g, little s. So this gamete from this cat is always going to be little g, little s, but this solid gray cat, depending on meiosis, can produce sperm and egg with any of these combinations. It can be big G, BS, big G, little s, little g, big S, or little g, little s. So you can produce 4 different genotyped cats. It turns out that 50% will end up being solid and beige, 50% will be solid and gray. So that is the answer. In sheep, the allele for black wool, big B, is dominant over the allele for white wool, little b. The allele for horns, big H, is dominant over the allele for being hornless, little h. Pure breeding horned sheep with black wool were crossed with pure breeding hornless sheep with white wool. So stop right there and try to write down what information you know. Right, it's a lot of words, so don't get confused by the wordiness, just write down what you know. Right, what are the genotypes of the parents? It must be big B, big H. Big H is the black wooled with horns. Little b, little h was the hornless sheep with white wool. What would the F1 sheep look like if they were crossed, if these two parents were crossed? So, if you got this black horned sheep can only give big B, big H sperm or egg. Let's say this is the male. So this male can only give sperm with big B, big H. This female can only have eggs with little b, little h. So, when they have babies, all of their children will be black and horned. Big B, big H, little h. Now I want you to figure out what the F2 generation would look like if 2 F1s were mated. So if the brother and sister were mated, not necessarily ethical in humans, but if these two were mated, so big B, big H, little h, what would you expect the F2 generation to look like? This, you should recognize, is a dihybrid cross. right? You're taking two, big B, big H, little h. So without even doing a Punnett square, you could be able to remember the 9 to 3 to 3 to 1 ratio and know that 9 out of 16 will be black and horned, 3/16 will be black and hornless, 3/16 will be white and horned, and 1/16 will be white and hornless. That's just knowing the ratio, 9 to 3 to 3 to 1. You can also do a Punnett square, right? We could use FOIL, like I mentioned, to figure out the different gametes. So the gametes can be big B, big H, big B, little h, little b, big H, or little b, little h. And then you would make a Punnett square showing the possible gametes on top for the sperm, possible eggs on the bottom, on the left, sorry, and then you would fill in all the possible genotypes. And if you looked carefully, you would count and see 9 to 3 to 3 to 1 ratio. You would get the following possible children. So this brings us to the second and final law that Mendel came up with, which is called the law of independent assortment. And this law states that the segregation of alleles for one gene does not influence the segregation of alleles for another gene. So in our case of our big R, little r, big Y, little y, what this is saying is that it's just as likely for the big R to go with the big Y as the big R to go with the little y is, right? One allele, R, doesn't have to do with the segregation of another gene, Y, right? Members of an allelic pair sort independently. So there's two ways to create diversity in meiosis in this case, right? We create a lot of possible combinations. Because at metaphase 1, we know that there's random orientation of homologous chromosomes on the metaphase plate. You can have both of dad's chromosomes and then both of mom's chromosomes separated, or you can have this alternative where you have mom's chromosomes and dad's chromosomes on top and the inverse on the bottom, which means in meiosis 1, you would split these into two haploid cells, and then in meiosis 2, you would get separate sister chromatids. And because of independent assortment, right, big R is just as likely to be with big Y as big R is with little y. So you're going to have an equal probability of having any combination, right? Because the R gene, the gene that encodes for seed shape, and the Y gene, the gene that encodes for seed color, have nothing to do with each other, right? They do meiosis separately. They assort independently. Right? So the alleles for P color assort independently of the alleles for P shape. So Punnett squares allow us to easily calculate the probability of seeing certain genotypes and phenotypes among offspring. So let's review some probability basics before finishing up this section. So if you flip a coin, what is the chance that you would land on heads? So I hope you would say C, right? If you roll one die, which has 6 sides, what is the chance that you would roll an even number? So for this, you would know that there are 6 sides to a die, 3 of which are even. So you have 3 out of 6 chance, which is 1/2, or a 50% chance of rolling an even number. So probability has a lot to do with genetics. So understanding these little things will help understand what we're going to learn about next, which is the product rule. The product rule states that the probability of 2 independent events occurring together is the product of their individual probabilities. This might sound complicated, but it's really not. If you want to figure out what is the chance of 2 things happening at the same time, you multiply the chance of one thing happening by the chance of the second thing happening. And that tells you if both of them will happen at the same time. It's also known as the AND rule because it's saying what is the chance of something happening, and something else happening. So for that, when you see this and this, you have to multiply the 2 probabilities. So for example, what is the chance of rolling a 4 on a die 2 times in a row. All right, if you roll the dice 2 times, what is the chance that you're going to get a 4 the first time and the second time? Right? That's the AND rule. So we have to multiply the 2 probabilities. What is the chance of rolling a 4 one time? That would be 1 out of 6. What is the probability of getting a 4 the second time? Again, 1 out of 6. Right? So chance has no memory, meaning every time you roll the dice, there's an equal 1 out of 6 chance of getting a 4. So the question asks, what is the chance of rolling a 4 on a die 2 times in a row? Meaning you get it on your first roll and on your second roll. So that would be 1/6 times 1/6. So the probability of getting a 4 on the first roll and the second roll would be 1 out of 36. All right, so that's the product rule. All right, so let's do it into practice. If you roll the die twice, what is the chance that you would roll a 5 both times? Pause here. The answer is A, 1 out of 36. Right, just like the previous slide showed. If you roll the die twice, what is the chance that you would roll an even number the first and the second time? So again, this is another product rule. So 50% the first time, 50% the second time. So that would give us C, 1/4 chance getting an even number 2 times in a row. So now let's try to apply it to a Punnett square. Consider a cross between 2 heterozygous individuals, big A, little a. What is the chance of getting a little a individual in the next generation? This is another way of saying, what is the chances of getting a little a from mom and a little a from dad? So you have a 50% chance, 1/2 chance from getting it from dad, and a 1/2 chance of getting it from mom, which means 1/2 times 1/2 gives us a 1/4 chance of getting a little a from dad and a little a from mom. So again, from this cross, big Y, little y, big Y, little y, this is from before, we know there's a 50% chance that the sperm will have a big Y, 50% chance that the sperm will have a little y. In terms of the eggs, 50% chance the eggs will have a big Y, 50% chance the eggs will have a little y. So this Punnett square is basically looking at the different probabilities. Right? So what is the chance of having a big Y? We're multiplying 50% chance times 50% chance. Right? Getting the big Y from the sperm and a big Y from the egg would give us a 1/4 chance of getting big Y. It's the product rule. 1/2 times 1/2 is 1/4. So each box represents 1/4. Right? Each box represents 1/4 times 1/4. So it's 1/4 of a chance. The other rule is called the sum rule, and that says that the probability of either of 2 mutually exclusive events occurring is the sum of their individual probabilities. This is a fancy way of saying, what is the probability that either event 1 or event 2 will occur? Right? What is the chance of this happening or that happening? This is called the OR rule. Instead of the AND rule, right, the product rule was the AND rule. What is the chance of getting this and that? You multiply. Product AND rule. The sum rule is the OR rule. Right? What is the probability of this or that? So what is the probability of getting a 3 or a 4 when you roll a die? So since it's a sum rule, we have to add the 2 probabilities. So you add the probability of getting a 3 plus the probability of getting a 4, and that'll tell you the probability of getting a 3 or a 4. So you would take 1/6 chance of getting a 3 plus 1/6 chance of getting a 4 equals 2/6, which is equal to 1/3 chance of rolling a 3 or a 4 when you roll the dice once. So pause here and answer this question. Just like we saw before, the answer would be B, 1/3. Again, looking at the same example from peas, what is the chance of getting big Y, little y offspring? Well, there's two possibilities. You can get a big Y sperm with a little y egg, right? A big Y sperm with a little y egg, that's 1/4. Or you can get a little y sperm with a big Y egg. That's also 1/4. So the chance of either happening, either getting a little y sperm or a little y egg, is 1/4 plus 1/4, which is equal to 1/2. And that, of course, represents the 1/2, or the 2 out of 4 that will be big Y, little y. So we're going to finish with this last problem. So dimples in humans are dominant, no dimples are recessive. Draw a Punnett square in your notes of a homozygous recessive male mating with a heterozygous female. So is the male or female true breeding for dimples? Which are the parental genotypes? What are the parental phenotypes? What percentage of their children will have dimples? Let's just stop there. So is the male or female true? So pause here and answer these questions. So hopefully you've had a chance to answer them. Is the male or female true breeding for dimples? The male is true breeding because he's homozygous, recessive. What are the parental genotypes? Well, we would have a homozygous recessive male would be little d, and the female would be big D, little d, since she's heterozygous and has dimples. Right? So the homozygous recessive male will have no dimples. The mother will have dimples. What percentage of their children will have dimples? So if you did a Punnett square, right, little d, big D, little d, you would see that half of their children would have dimples and half would not. So 50-50. What is the chance of having a girl with dimples? So this is where we have to use the product rule. So the product rule says, what is the chance of two separate things happening? You just multiply the separate probabilities. So what is the chance of having a child with dimples? 50%. Right, according to this. 50% will have dimples. What is the chance of having a girl? Again, 50%. So we multiply these two probabilities, right? So it's 1/2 times 1/2 is equal to 1/4. Right? 1/2 having of dimples times 1/2 having a girl, and that'll give you 1/4 chance of having a girl with dimples. Right? She's a girl and she has dimples. What is the chance of having a heterozygote or a homozygous recessive child? So for this, we have to use the sum rule. So what is the chance of having a heterozygote? Right, 50% chance, 1/2. What is the chance of having a homozygous recessive child? Still 1/2. So in this case, 1/2 plus 1/2 equals 1. So you're guaranteed to have either a heterozygote or a homozygous recessive child. There's no possible way to get a heterozygote, a homozygous dominant child in this case. If two parents are heterozygous for 3 genes, big R, big Y, big T, little t, what is the probability that their child will also be big R, big Y, big T, little t? So this is tough. This is a tough question. So try to pause here and see if you could come up with the answer. You might have been able to figure this out, but you can use the product rule to simplify this problem. So the probability that an offspring inherits that genotype is the probability of having big R little r times the probability of having big Y little y times the probability of having big T little t. So what we could do is we could construct separate Punnett squares. We can have one for the big R little r times the big R little r, one for the big Y little y, and one for the T's. And then we're trying to figure out what is the probability of getting offspring with this combination of alleles. So let's just look at the R first. Well, there's a 50% chance, or 1/2 chance, of getting big R little r. If you look at the Y Punnett square, big Y little y, 50% chance of getting that big Y little y. 50%. Again, here's big T little t, 50% chance of getting big T little t. So what is the likelihood of getting big R little r and big Y little y and big T little t? 1/2 times 1/2 times 1/2 is equal to 1/8. So that is the answer, is 1/8. So pause here and try to figure out the answer to this question. It's the same type of problem. And the answer is also 1/8, right? You would see that 50% would be homozygous recessive for each of these. So it's 50% times 50% times 50% would be 1/8. Phenylketonuria is a recessive disorder. If a man with PKU had a child with a healthy non-carrier female, what is the probability that their first child will be born with PKU? So let's try to figure this out. We have a man with PKU, which is a recessive disorder. That means he must be little p, little p. And again, you can pick any letter you want for this. It'll give you the same answer. And he has a child with a healthy non-carrier female. Means she has to be big P, big P. That means all the children will have big P, little p, which is healthy. Meaning this is A, 0% chance that the child will be born with PKU. If a man with PKU had a child with a female who is a carrier, so that's little p times now big P, little p, we would see that 50% of their children would have PKU. So the answer is C. Again, let me just have a note about probabilities. This is not to say that if they have 8 children, 4 will have PKU and 4 will not. This is probability, right? So that's like saying if you flip a coin 8 times, it is most likely that you're going to get heads 4 times and tails 4 times. It's possible that you're going to get 8 heads, right? We're just talking about probabilities, not actuality. Finally, here's another question about PKU. If a man with PKU had a child with a female carrier, what is the probability that their first child will be a male born with PKU? So this is multiplying the 50% chance of having PKU with a 50% chance of having a male, giving us B, 25% chance of having a male born with PKU. So that is the end to part 1. Make sure you understand that stuff very well. Go through the practice problems. I would even watch this whole first half again before moving on to part 2.